So good morning, United States. Good afternoon, Europe, and wherever else you are in the world. I welcome you to today's Fine seminar series. My name is Carsten Schradin. I'm a Director of Research in Strasbourg, France, and it's a pleasure for me to host today's Fine. I have one short announcement to make. We hear from many people that they appreciate we have this Fine seminar and how thankful they are. And I have to say, you don't have to be thankful because it's totally egoistic. We do this because we love to meet interesting people once a week. And if you also like this, then it's of course great. It's some kind of mutualistic cooperation. The only thing where I'm a little bit altruistic is that I took over the job to write certificates for students that participate to Fine so they can get credits. And for this, I put up some rules at the very beginning, which is you should send me the summary directly after the Fine or earliest Wednesday morning by 6 o'clock tomorrow morning, because that's when I drink my coffee. And before I work for my students, I put everything into the Excel list. And I have to say that I will strictly stay with this rule. And if you send me your summary much later and you have lots of excuses because you have something more important to do instead, then I'm sorry, I also have. And I will not send 20, 40, 50 emails discussing this. So just to make this clear, the rule is you should send it to me after the Fine or tomorrow early morning, and I fill it in. And if it comes later, I will just ignore it because I have my own students. I have to invest time into this. And I hope that everybody accepts this, and that because of this, we can continue with this also with the next term, so that the next students can also benefit from getting such a certificate if they can need it. And by the way, I'm German, I'm very direct, so I say what I think. I'm sorry if anybody thinks that's rude, but I just wanted to make this clear. So today we are going to have a fascinating talk by Adriana Maldonado-Chaparro, who is at the Universidad de Rosario in Colombia, about social and demographic consequences of changing environments. Adriana, she studied in Colombia at the Universidad Nacional de Colombia. She did her master thesis about, of course, some of the most interesting animals on the world. That is social rodents, and the capybara, actually the largest rodent in the world. And afterwards, she moved on to do a PhD with Dan. Now I never know if it's Dan Blumstein or Dan Blumstein. I guess it's American, his last name. Sorry, Dan, if you're here, that I pronounce it wrong. Working on this famous project with the marmots in the United States, based at UC, University of California, I think it's Los Angeles. And afterwards, she moved on to the Max Planck Institute in Konstanz, which is actually only 2 hours drive away from where I'm based at the moment, to work with them in Farine, especially on zebra finches and social structure. And I think we will hear a lot of this today. Adriana has a great reference of publications. She already published while she was a master student. And what is always interesting to see is that over the years, there has been a steady flow of papers coming out, and the quality of these papers has always been increasing, showing that it's really also her own work and her own expertise that goes into this. And it's very nice and fascinating to see how such a career is building up and how there is progress, and it's getting more and more interesting. And since 2020, she is a professor back in Colombia, the Universidad del Rosario, and she's going to work there. There has, of course, like for most people, some interruption because of the corona crisis. She's going to work on these fascinating birds, the so-called all birds, still in collaboration with the Max Planck Society in Germany, and especially in collaboration with the Max Planck Institute for Animal Behavior in Konstanz. Also important to tell everyone, you know that at the Fine we have our own homepage. I will put this, that was wrong. I will put this once again into the chat for everyone, that you can find the homepage. It's always difficult to find the chat function when one is sharing. I will put it later when I'm less nervous. And we have this function here that fine presenters can send us teaching slides before or soon after or long after they presented. And Adriana already sent me her slides, which you can download here. It's a very well done and interesting summary of some of her most important results on environmental disturbances and social instability. So we welcome everyone from the fine audience to come to our homepage and download these and other teaching slides if you're interested to use them within your curriculum of teaching animal behavior at your own university. Yeah, with this, I'm finished with the introduction, and for somebody who has been studying social organization for many years, who's now moving with one species at least to more social structure, I'm very much looking forward to your presentation, Adriana.
Well, thanks, Carson, for such a nice introduction. I have to say I'm impressed with, with it as well. And thanks, everyone, for attending the talk today. Well, as Carson said, I will talk to you about ecological and social responses to changing environments. And when looking at this picture, I just want you to imagine some of the first impressions that shape and inspire some of the questions that I've been addressing during my work as a biologist. So this is Casanare. This is part of the eastern savannas in Colombia. It's a gorgeous ecosystem with a huge diversity of plants and animals. And most importantly, it's a highly contrasted seasonality driven mostly by the patterns of precipitation. And so when you're there and it's raining, the savannas are flooded and you see green everywhere. Plants grow and there is plenty of food available for all the animals. And I have to say, there are actually very few places where you can stay dry. But when there is no rain, everything changes and the savanna dries out and there is little food available and the temperatures are high and it's very hot, and overall the environment creates this harsh habitat to live in. And as you can see from these photos, both taken from the same spot, just different times of the year, the landscape is very contrasting between the rainy and the dry season. And when you put this and also observe the animals that live in there, you can start thinking about how are they able to cope with these changes. And particularly, I started wondering about how and why some animals were able to cope better than others. And when looking at the capybaras, I started wondering about the potential role of groups in helping individuals to adjust to these changing conditions. So from an individual's perspective, the environment can change in sort of predictable way throughout the year. For example, as I just mentioned, in tropical areas, we can go from a wet season to a dry season. And in temperate zones, probably the ones that we are mostly related to, we go from winter to spring to summer and fall. And because these changes within the year occur with some predictability, the result is that there is less uncertainty in their occurrence, and individuals somehow know, or they have a better expectation of what to expect in the near future. However, environments can also vary from year to year. For example, in the amount of food available for foraging, it may not be as high in one year compared to the previous or to the next year. And this variation among years is less predictable than the within year variation. So from one year to the others, individuals experience less predictable changes in the environment and with increased uncertainty. And in addition to this, environments can also vary in much more less predictable ways. And this is the case of extreme events, such as fires, hurricanes, storms, and heat waves. And we could refer to these ones as disturbance events. And these extreme events can occur naturally, but they can also occur as a result of human activities. And what is important from these extreme events is that they occur with even less predictability. So animals experience even more uncertainty about the future environmental conditions. One thing that we can worry about is that recent climate studies suggest that the length and the frequency and the intensity of these extreme events is increasing at the global scale. So here, Mire and collaborators showed the propensity or the probability of extreme events over the years. And when we combine these with global warming temperatures that we already know that they are happening under any of the scenarios that they have explored, what we see is that the environment is changing at faster rates and in less predictable ways. And the second thing that results from these changes is that more species are experiencing conditions outside the historically experienced. This we often refer as novel environments. So this unprecedented change is posing a threat to biodiversity. And even though some species are responding, others are threat to local or global extinction, with some groups here being more susceptible than others to these extinctions, as shown by Bellard and collaborators. So because of this, I think that the question of how animals respond to fluctuations in environmental conditions becomes even more relevant. And today I want to address this question from different perspectives. First, at the individual level. So we know individuals differ in different characteristics, such as the age, sex, their physiology and behavior, but they also differ in the way that they can respond to the physical environment. And I will make here a note. I will refer as two different types of environment: the physical environment that includes factors such as the climate and food availability, and the social environment that includes all the potential individuals with whom the focal individual can build relationships with. But it's clear that within populations, not all individuals interact with each other. So at least in group living species, there is a structure that is mediated by the social groups. So I will also explore the group level response to environmental variation, as well as the population level response. And I hope that during this talk, I can show you how groups are an important component understanding the responses to environmental variation and how they can mediate the effects from individuals to populations. During the talk, I will also refer mainly to two study systems. The first one, this is the yellow belly marmot system. With this system, I will explore responses to historical environmental conditions. And I'm somehow lucky that Dan Blumstein already introduced this system in his talk during the last series of this seminar. So here, I will only go through some key points of this species. So first, the study site is located here in Rumble in the Western United States. Rumble is short for the Rocky Mountain Biological Laboratory in the Upper East Valley in Colorado. The population is distributed between 2,700 meters and 3,100 meters above sea levels. And the individuals are distributed in colonies, here represented by the red dots, and satellites represented by the black dots here. The colonies are groups that consist of one or several mature lines that do not go extinct over the years, whereas the satellites are usually composed by a single mature line that can go extinct from year to year. So the yellow belly marmots, this system is one of the longest running rodent research projects in the world. The population has been studied since 1962, And since 2002, the social behavior has been systematically recorded. So the marmots are diurnal, they are facultative social rodents. They hibernate quite a long time, 7 to 8 months, from late fall to early spring. And then when they are active, they need to first reproduce and then gain enough body mass so they can survive hibernation. And so every year during this active period, we trap all the individuals in the population. If it's a new individual, we give them an ear tag. And then we also give them a 4 mark here with 4 dye for observations from afar. And for each individual, we also follow their reproductive outcome. During the active period, we also follow their behavior, especially we follow the social behavior where we systematically collect the affiliative and the aggressive interactions, and we keep track of who initiates them, who receives them, and the locations in which they happened. So the first question I want to explore with this system is if individuals exhibit plastic responses in their morphological traits, and if this happens, then if the response vary among individuals. So let me provide a little bit of context here. So it's been proposed that phenotypic plasticity allows organisms to cope with environmental heterogeneity. But this can only happen when environmental cues about the heterogeneity of the habitat are fairly reliable. So under these circumstances, individuals can rely on these environmental cues to predict the future conditions. And here I use the term plasticity to define the ability of an individual to modify its phenotypic state or activity in response to variations in environmental conditions. And we also heard a lot about phenotypic plasticity and all the different connotations it has in the 2 recent talks by Dustin Rubenstein and Barbara Taborski. So I'm glad I don't have to go very deep in this topic either. So second, it has also been proposed that under current conditions of climate change, phenotypic plasticity provides a potential for individuals to respond rapidly and effectively to environmental change. And this has been shown in a wide range of species, and here I'm just going to show 3 examples. This is the gray tits, the collar flycatchers, and the pit flycatchers. In these populations, the mean laying date has advanced in relation to warmer spring temperatures. So seeing that basically individuals or populations can track changes in the environment. But how does it relate to marmots? Well, in the Rockies, where marmots live, spring temperatures and precipitations vary considerably from year to year, and spring temperatures are getting warmer, as we can see in the green line, and precipitation is slightly decreasing over time, as seen in the blue line. In addition to this, we also know that marmots at Rumble are emerging earlier from hibernation in response to warming spring temperatures. And this has been happening since around 1970s, as reported by David Inouye and collaborators in 2000. So based on this, we would expect other phenotypic traits to also respond to varying climatic conditions. And with this in mind, I evaluated the plastic responses in body mass after hibernation. So this is the June body mass in response to spring temperatures. And the main aim of this study was to identify the patterns of plasticity and to determine if there were individual differences in the plastic response. So here I'm showing the response of body mass. In the x-axis, we see the spring temperature, and the y-axis, the body mass. So in the graph, the red line indicates the population response, and the gray lines, the individual level response. What we found is that marmots that emerge in warmer spring temperatures have larger June body mass. So this indicates that plasticity plays an important role in the short-term response of the population to changing environments. In addition to this, we also found a significant difference in the degree of plasticity among individuals, meaning that some individuals, and here I highlight two of them just for as an example in blue and purple, meaning that some individuals are more plastic than others. And this suggests that plasticity contributes to the maintenance of individual phenotypic variation in the population. Also, and we see and we show that plasticity affects the distribution of the trait. So there is a narrower range of trait variation in colder environments compared to the range of variation in warmer environments, which is larger. And this pattern suggests that the climatic fluctuations have led to important phenotypic changes in the distribution of traits in the population. But why is studying June body mass very important or so important in this population? So it's very likely that the early season factors can influence and determine the late season responses. And so here I show how early body conditions influence body mass before hibernation and reproductive outcomes. Again, in the x-axis, you see the body mass in June. And on the left, we see how heavier marmots in June have larger body mass during the late season. And this is basically the body mass before hibernation, which is important for overwinter survival. On the right here, we see that larger marmots during the early season are able to win larger litters. And so if body mass in early season can respond to these changes in the environment, and it can also influence reproductive outcomes and the body mass before hibernation, then the next question that I posed and I asked myself was what was the contribution of these changes in body mass and the demographic traits and environmental conditions to fluctuations in the population. And to study this, I worked in close collaboration with Dylan Shields from University of Sheffield to develop the population model that I will present in a moment. To give you the whole picture of what's happening with the marmot population, here you see a quick overview of the trend since 1976. So we can see that historically, the marmot population fluctuated around carrying capacity, and then around the year 2000, it threefolded in numbers until it crashed around in 2010. So back in 2010, Osgood and collaborators proposed that this increment in numbers could have been caused because an increase of the end of body season, because of an increase in the end of season body mass of the individuals in this population that allowed them to enter hibernation in better body condition, and therefore they will have lower overwinter mortality. So if we put this together with my previous results, this indicates that body mass changes can contribute to the variation in population dynamics. So here, the aim was to quantify the contributions to population growth. So to do this, we built an environmentally driven integral projection model. Here, I just present the life cycle of the marmot, where I use the body mass as a continuous trait to describe the population stage-dependent demographic rates. So in this diagram, we can see the census points where the number of individuals at the time t and the time t+1 depend on the size of the animals at that time. We also see that individuals can survive with a probability based on their body mass, and during the summer they can reproduce based on a probability that is also driven by their body mass. Within that time, individuals grow. So they gain or they lose body mass. And this growth, we split it in two terms. First, the growth during the winter, which is basically during hibernation, and then the growth over the summer, which is during the active period. So we call this the winter growth and the summer growth. In addition to this, I use the winter temperature, the spring temperatures, and snow-free days as the environmental drivers of population fluctuations, because we know that these variables can affect the body mass. So the environmental factors can affect population dynamics directly through the effects of survival or reproduction, and indirectly through the effects of body mass. So trait-mediated effects. And here we aim to quantify these effects. In this plot, what you see is the contribution of the trait-mediated effects and the demographic traits to the fluctuation on population growth. So on the x-axis, we have a measure of the contribution to population fluctuation. And on the x-axis, on the y-axis, we have the different demographic traits. To interpret this, the longer the bar, it indicates that we have a larger contribution. So what we see here is that survival and reproduction are the largest contributors to variation in population growth. And when we look into growth, so the trait-mediated trait, what we see is that contrary to what we were expecting, we showed that even though we have an evident influence of the environment on body mass, changes in body mass have a small contribution to variation in population growth rate. We also show that this contribution or this effect has a 1 year lagged effect. So this means that the effect of a good or a bad year on body mass will not be evident until the next season. And this suggests that marmots exhibit a mechanism of growth compensation by which individuals can emerge from hibernation in poor conditions, and then they can catch up during the summer. But so populations, and this is what I called at the beginning, the indirect social environment, fluctuates over time. And these changes at the population level can also drive changes in their direct social environment. That is, the groups can also vary due to demographic events such as mortality, natality, or dispersal. And so individuals are then expected to adjust their behavior to these new social conditions that they experience. So for example, they can modify their decisions about how they interact or who they interact and how often they interact with. So if the social environment can also fluctuate, then the next question is how individuals or members of the group adjust their behavior to these changes. To explore this, we used the observability interactions of marmots to construct non-pop female social networks. And here I present 4 networks corresponding to the same colony. This is Marmot Meadow in 4 different years, 2006, 8, 10, and 12. The dots in each network correspond to the nodes of the network and represent individual females. The lines correspond to the edges of the network, and they represent the presence of an affiliative interaction. So what we see here is that the size of the female groups vary among years, as well as the structure of the social relationships. So to characterize the social structure, we use several social attributes. The first of them, so we characterize how sociable was an individual using out degree, how likable using in degree, how closely associated to other individuals using closeness centrality, and how important an individual is within its group using betweenness centrality. So here in these graphs, they represent the summary of the results. We'll see always in the x-axis, the group size, and then on the y-axis, the trait that we are evaluating. The continuous line represents the regression on the observed values, and the dotted line represents the regression on the values obtained from the randomized network. So what we see is that overall, the social attributes vary in relation to group size. And specifically, as group size increase, marmots are less likely to add new partners to their group, to their social network. And they are also less likely to receive new social partners. And when we look at the closeness and betweenness centrality, they also decrease as with increases in group size. So this suggests that there are individual level mechanisms that allow marmots to balance the cost and benefits of maintaining social relationships. And so, well, we know that the social environment can influence the expression of life histories and can also have fitness consequences. So the next question that I asked was about the potential fitness consequences of social relationships. And probably you're thinking already, well, it's well established that social relationships provide benefits. And here I'm showing some of the studies that show a clear link between social bonds and demographic parameters. So in the yellow baboons, the offspring of females who had stronger bonds have higher survival compared to the offspring of females that have weaker bonds. And in Chekma baboons, the offspring of females who form strong bonds similarly live longer than the ones, the offspring of females that form weaker bonds. And these species, females that had stronger social relationships also live longer compared to the ones that have weaker social relationships. And when looking at the marmots, we saw that this was not the case. And for marmots, being social can have negative effects as it can reduce the probability of survival. So here in the x-axis, we see the strength of the social relationships. And on the y-axis, we see the probability of surviving over winter. So in marmots, the more amicable individuals are less likely to survive over winter, suggesting that there is a cost to sociality. So to wrap up this first part of the talk, we saw that climatic fluctuations have led to important phenotypic changes in the marmot population, that marmots exhibit different levels of phenotypic plasticity that contributes to the maintenance of individual phenotypic variation in the population. Body mass dynamics, it has a small contribution to variation in population growth. Marmots can adjust their social relationships to changes in group size and composition, and also affiliative social relationships can have a fitness cost. So up until here, I have explored the responses to historical fluctuations, but as I mentioned at the beginning, we expect that the frequency and intensity of extreme environmental conditions to increase. And so now I will explore how groups respond to disturbance events. And for this, I will only focus at the group level, on the group level responses. So there has been already some studies that capitalize on natural extreme events, and it has been shown in at least in 2 species that disturbances such as hurricanes and fires can alter the social structure of a species. So for example, Testard and collaborators recently showed that macaques can build new social relationships after a hurricane. And in fairy wrens, the birds that live in habitats that were affected by fires had stronger social relationships compared to those that live in habitats that were not affected. So if these extreme events can affect the social relationships, one of the questions that just comes to my mind is how these disturbances can affect the performance of the group, or how can we measure the cost of the perturbation to the system? And to tackle these questions, I use a different study system. This is, here I use zebra finches. In the wild, zebra finches live in a somewhat unpredictable environment, also driven by the pattern of rainfall. They live in large colonies of hundreds of individuals. They are highly social and they form stable pair bonds. But the studies I will refer to here happen in captivity in indoor and outdoor aviaries. And the first question that I want to ask is, what is the cost of disturbance events to group performance? So to explore this, I designed a disturbance experiment to evaluate the influence of repeated disturbance events on the ability of a group to perform a task. And I also explored the potential underlying mechanisms that are involved in this response. So for this experiment, we developed an automatic tracking system that relies on backpacks. Here is the zebrafinch with the backpack with a QR code, which is this white square that you see here, that can be automatically read by cameras and then translated by the computer. So here you see a video on how it looks, a foraging task, and how the system reads the identity of each of the birds in the frame. What we measure as group performance, so we use foraging efficiency as a measure of group performance. And this corresponds basically as the amount of zucchini, here the centerpiece, eaten by the group in a 45-minute trial. So in this experiment, we used two flocks, one that had been together in the same groups for 6 months previous to the start of the experiment. And we will call this part the stable period or the pre-disturbance. During this time, we will measure the baseline foraging efficiency of the flocks. Then we created the disturbance period, and this consisted of 3 disturbance events in which we split and recombine the flocks. So when we split the flocks, we captured all the birds from the main aviary, and we divide this in 3 subgroups composed by randomly selected individuals. Then each of these, one part will go back to the main aviary, and then the other 2 subgroups will go to different rooms. And we kept them in different rooms for 2 days. After these 2 days, we captured again all the individuals and we brought them back to the main aviary. At this point, we will measure again foraging efficiency for 3 days. We did this 3 times, and then after this, we stop the manipulations. We call this the post-disturbance period. And then we measure again once after we stop all the manipulations, and then again 3 weeks after the foraging efficiency of the flocks. What we found here is in terms of the foraging efficiency that compared to the pre-disturbance event, the foraging efficiency during the disturbance showed a significant decrease. And when looking at the post-disturbance period, we saw that this efficiency was not recovered to initial values, even after 3 weeks of no disturbances. When exploring at the potential underlying mechanisms, we saw that the social relationships were affected by the repeated disturbance events. And so here, I show the association strength calculated from the foraging association networks. So low association values indicate weak relationships, and high association values indicate strong relationships. And what we found is that compared to the pre-disturbance period, in the disturbance period, relationships were weaker compared to the pre-disturbance. And then even though they show a tendency to recover, they never went back to initial values. This pattern, it's more clear when we look at the coefficient of variation. So here, low values indicates that the strength of social relationships is homogeneous among individuals, whereas higher values indicate a larger variation in the strength of the association among individuals. So this can be interpreted as higher exclusivity in the relationships. So our results indicate that compared to the pre-disturbance period, the coefficient of variation increased, indicating that the relationships are more exclusive, which means that probably strong relationships are getting stronger and weak relationships are getting weaker. This, same as before, there was a tendency of recover, but they didn't go back to, the values didn't go back to the initial values pre-disturbance. So together, these results suggest that groups may not be resilient to disturbance events, at least in the short term. So if the pattern of social relationships are important, then I ask if stable social relationships provide some fitness advantage. And for exploring this, basically, we took advantage of the fact that zebra finches form distinguishable pair bonds. So we set up single sex groups. So we raised individuals in groups of the same sex until they were adults. And then we created these 10 replicated mixed groups with 14 females and 14 males. Males and females totally unfamiliar with each other. And once we put them into the aviaries, we monitored all their social interactions among all individuals in each of these aviaries or colonies. Again, here we use our automatic tracking system to identify the position identity and the identity of each of the birds in the colony. So we monitored from the point in which we put the individuals in the aviaries and long enough to allow individuals to form a pair bond. It was about 30 days. From these videos, we used the clumping behavior to quantify the proportion of time that two individuals were detected in body contact. So here we see an example of two birds clumping, and you can see that they are in body contact. And from these, we calculated the simple ratio index, which we use to measure bond strength. With this, we built daily social association matrices that then we transform into focal daily association matrices. So this means that for each female in the colony, here I'm going to call this female the focal female, we describe the daily association strength with every other male in her group. So male from 1 to 14, and for each day, we describe how the strength of their relationship developed through time. So what we found is a wide range of variation in the patterns of pair bond formation. And here I'm just showing an example of a pair that form a stable and a strong relationship since very early in the experiment. So this means basically that the female showed a rapid differentiation in the strength of the association with a particular male. But this was not always the case. Other females, they didn't show this clear pattern, and there was no preference in their association. So you see here that their relationship is not as stable as the one that I previously showed. What is important is that these pairs that form stable social relationships were more likely to initiate a cluster, they were more likely to initiate a clutch faster compared with the pairs that form less stable relationships. And this indicates that the stability of a social relationship has a reproductive advantage. So far, I've shown that social instability caused by repeated disturbance events can affect group performance, as shown by a reduction in the group foraging efficiency, that the underlying mechanisms that mediate changes in group performance are likely to be related to the pattern of social relationships, that groups may not be as resilient to repeated disturbance events, at least in the short term, and stability of the social relationships can represent a reproductive advantage. So to wrap up, I propose that the social relationships are a mechanism by which groups and populations can respond to disturbance events. But when these relationships are disturbed or disrupted, the system can enter in a state of social instability that can affect different components of the social system and can have consequences at different levels of organization. So if these social relationships are a key mediator for population dynamics, so it becomes important to understand at different levels the consequences of social instability. So for example, asking at individual level, the physiological and fitness consequences, at the group level, the effects on other components of the social system, and at the population level, the effects on population persistence. And to start working on this, on understanding the consequences of social instability, the first step is to build a conceptual framework. First, to understand what does it mean, social instability. So we start working on this in collaboration with Lawrence Hayes and Louis Ebernsperger. And so far, we try to unify the concept of social instability. And second, we try to quantify at different levels the effect of social instability using a meta-analysis approach. More on this, hopefully soon. So here I proposed, and I think that the social structure should be placed in a central position to better understand the population and also, why not, the community level consequences of unpredictable changes in the environment, particularly those resulting from anthropogenic activities. So on one hand, maybe we could identify behavioral mechanisms that can be used to predict responses to rapid environmental change. So from group dynamics to populations. And on the other hand, by integrating into our framework the different feedback loops among different levels. So as we propose in our recent review, where we highlight that individual behavior can define the direct and the indirect environment, social environment, and that this environment can in turn feedback and shape the different selective factors that can drive individual behavior. And with this, I would like to thank all my funding sources, as well as my PhD lab, Bloomstein's lab, my postdoc lab, Farine lab, and my current university, Universidad de Rosario, where I'm starting my research group. Thank you.
Thank you very much for a great talk about past, current, and future research. It's really stimulating. To tell everyone how it is like it always goes. If you have a question, then please type a question mark into the comments field. I will then call you up one by the other. When I call your name, then still please quickly introduce yourself, say what is your name, where you're coming from, maybe what you're working on before you ask your question, so we know who we are here talking with. For the people that are on YouTube, at the moment there are 25 people watching on YouTube. If you do have a question, you can write it in the chat function there. I will once in a while have a look at the chat in YouTube. And if there are questions, I will bring them up here in the audience. So as there is no question yet, there's one coming in. Then the first question comes by Kaya Tombak.
Hi there, thank you so much for that great talk. I was wondering about the sort of quantity versus quality of relationships, and how you found that in the marmots, it seemed like the quality could actually be a cost to fitness, but then the quality of a relationship might be important for things like zebra finches. What do you think causes that discrepancy? And is there something to do with, I guess, whether sort of survival or reproduction might be more important to the given animal? So, you know, if overwinter survival is more important, maybe you need just more group mates, more than you'd need strong relationships, for example.
Yeah, great question, Kaya. Thanks for asking that. Indeed. So I would say that the main difference between these two systems is marmots are facultatively social. So it's, you know, they socialize because they sort of have to. And so one of the explanations that we think it's valid for the results we've got here is that marmots hibernate individually. So these marmots that build strong relationships during the summer, we think that they might go into hibernation together, and this might disrupt the cycle of hibernation and they wake up more often, or we don't know exactly how it will disturb the hibernation cycle, but we think that it might create an energetic demand, and that's the reason why it decreases or it negatively affects survival. It is different from the hibernation in, say, the European marmots, that they do hibernate socially and they require being with other individuals to better cope with hibernation. So those are two different things there. And you're right in terms of like what it means to have a good relationship in one system might not have the same effect or consequence for another system. So in zebra finches, and that's why I also, in this case, we target a specific relationship, the pair bond, which is very important for reproduction, right? Having a good pair bond means, at least from our studies and other studies, not based necessarily on the type of approach we had, that has reproductive advantages. So it has a fitness consequence to have a good partner around you. So, yeah.
Thank you so much. And I forgot to introduce myself, Kaya Tombak, postdoc at Hunter College. Thanks for the talk.
Yeah, thank you for the question, Kaya.
Then the next question is by Eduardo.
Thank you. Thank you, Adriana, for a wonderful talk. I'm Eduardo Fernández-Duque from Yale University in the US. And I have a rather technical question. I was hoping that you can share some of the specifics that have to do with the devices you're attaching to the zebra finches to more closely monitor. So for some time, I remember it was several years ago that we were discussing this with Tony Di Fiore, with whom we were considering how to better monitor, not to better monitor, to differentially monitor the interactions between the pair bonded TDs and owl monkeys in the field. And we never did it for lots of reasons. So, but again, we're thinking about it for owl monkeys, and I'm very, but I haven't caught up with the technological development. So I was wondering if you could comment a little bit or point me at who may help in understand where we're now with distances at which they will monitor. I mean, the things that are crucial to implement them if you're going to do it in the field. Specifics on how long they last, how you download, at what distance, what's the situation with those these days?
Okay, that is a challenging question. Okay, in terms of this specific automatic tracking system, I think it's hard to implement it in the wild. It works pretty well in captivity, and indeed we have that running in a different setup, also with several finches, but monitoring a larger group, like 80 individuals. In my experiment, we were monitoring 28 individuals, and so you could go to even more. And if I understood correctly, you want to like sort of understand a little bit more about like how these devices work, right?
Yeah.
So this is basically like a barcode, like the ones that you probably use in the supermarket, that they just go and register what it is. And so we have a Raspberry Pi with a camera that is taking photos, and it can also take videos. It depends on how you set it up. And it takes, in my case, we took photos every 3 seconds. And then we process that using an open source software that is called Pinpoint. And so what the software takes out is basically the identity of the individual and the spatial location, and also the directionality of the code. So in my case, I didn't use the directionality because birds could be very easily, turning their heads, and that doesn't mean that the code is actually reflecting what they do with the head. So I only use body contact, and I measured using the photos, you basically measure the distance between individuals. So you can get a sense of what number of pixels correspond to a body contact, for example. And then I also did this for being in close proximity. So 1 body length between 2 individuals. So this is a little bit of like extra work standardizing these measures and how it corresponds to number of pixels. Then it's a matter of going in through and building scripts to quantify this using social networks. But if implementing that on, say, your system, that will be a different approach. And we tried this in dolphins in Brazil. So there is a colleague of mine here at the Max Planck. He works with cooperative behavior between dolphins and fishermen. And what we tried to do is to put a QR code on the hat of the fisherman. And then using drones, we would look at the location of the fisherman. And well, the dolphin like bubble, you could infer from the video footage of the drone. But this turned out to be a little bit more complicated than we would expect. But I think it could be doable using different devices, right? So maybe not the same Raspberry Pi camera that is fixed in a single point, but something more movable. Yeah. I don't know if I answered.
Oh, you did. I'll ask you some more questions later, because I think it can help me get started thinking more seriously about it, both in captivity and in the wild. Thank you.
Okay.
Then the next question is by Gloriana.
Hi, hola Adriana.
Hola.
Adriana Maldonado-Chaparro from the University of Costa Rica, and I work with communication, social behavior in bats. Thank you for the talk, Adriana. I enjoyed it. So I had 2 questions, but I'm going to just ask one first now. So you were looking at the effect of sociality on survival in the marmots. And from what I understood, you only looked at winter survival. So is this a normal proxy that you use to measure survival? And what effects could this particular measure of survival may have on whether other patterns of survival may be better represented by sociality? For example, you're saying that normally the marmots in winter, like they have different patterns of social behavior that they may have during the summer, for example. So could this measure of survival, winter survival, affect other things that you may be looking at by looking at survival in other times and the effect of sociality on that?
Okay, thanks for the question. So winter survival is perhaps the key component of that drives variation in marmots populations. So we could also look at summer survival, but that's quite harder because we would have to disappear from our population during this active period. And individuals also disperse during this time. And so we won't be able to properly tell apart which individuals died and which ones dispersed. And so we would overestimate the survival during the summertime. So that wouldn't be a good proxy if we want to look into that. We could, I mean, we do have studies looking into dispersal decisions and social relationships. So Tina Wei in 2008, I think is her paper, she looked into betweenness and dispersal. So marmots that are more embedded in the network, they tend to disperse less than marmots that are in the, let's say, peripheral part of the network. So we've looked into different proxies of fitness and different social attributes. But yeah, the one that I was interested was in the survival, winter survival specifically. Yeah, this is also because, so we have two different time frames, right? The summertime where all these relationships develop, and then the winter time. So how this translates from summertime into what happens during the winter and how it results in the next year in the population. Yeah.
Okay. Thank you.
Okay. Thanks for the question.
Thank you. Then I have myself a question, and then there's a question from YouTube. So my question is, it mainly shows how little I know about social network analysis, even though I should know much more, as I have real experts working on this in my own team, and I'm also interested in this. So with these marmots, you showed that with increasing group size, all the measures you had decreased. It was how sociable they were, the out degree, how likable they are, the in degree, how closely they associate, the closeness, and the betweenness. So just for my understanding, does this really mean in larger groups they get more unsocial from their social structure? Does it mean they distribute it to more potential social partners?
Yeah, that is indeed. So thanks for asking that, Karsten, because I think that's, I tried to address that also based on a question that I got before, but seems that okay. So as you get more individuals in the group, it's not that marmots are isolating from the group, it's that they just don't build more social connections with other members of the groups that are joining. So they just tend to keep their old associations and do not add any more. That's what the study is showing, or the analysis is showing. And also they don't do it like the out degree, so they don't go out and trying to find more connections, but they also don't tend to receive any other. So if someone wants to be their friend, they are not likely to get a new one.
Okay, so they're quite kind of socially buffered. It doesn't, changes in group size doesn't change too much for them. Then there is a question from YouTube by the name is, it's the full name, so I don't know what his or her name is. Y Crow. He or she says, thanks for your nice talk, Adriana. Now it comes. This is Claudio de la O from EU Latina in Mexico City. My question is, did you quantify allopreening among zebra finches? If yes, how much your social bonds measure? How did your social bond measures change? I'm not sure what is allopreening.
Yeah. Short answer is no, not directly. The long answer is so often when two individuals are allopreening, they are in very close proximity. And what I was measuring was individuals that were in body contact. So if they were allopreening, I would capture them in the picture, or with a device. So indirectly, the allopreening is in there, it's just not targeted to that because I don't have the means of identifying the detailed movement of the individuals, like actually printing the other individual in the pair. So I hope the answer is clear.
Okay, just because there's maybe other people that are not native English speakers. Alloprinting, I just tried to Google it. It's like grooming in mammals.
Yeah, so alloprinting, it's just like, so birds, one of the behaviors that they use to strengthen their social bonds is by cleaning the feathers of the other individual. So we use it in the same sense as in mammals. So.
Okay. Then we have a question by Ilvana.
Hello, everyone. My name is Silvana Salas. I am PhD student in Chile. Hello, Adriana. Thank you for your talk. It was nice and very clear. I have a question about the body size, because you said that body size seems to determine the groups and population dynamics, then because of demographic and reproductive features. So you also said that the body size seems to increase when the temperatures are higher. And I was wondering if you have looked about why is this happening? Why is temperature producing differences in development? Maybe thinking about energetics or the way that resources are being used to stay warm or increasing in size, or why is this happening?
Yeah, thanks, Ivana. Okay, so the body mass that I measured was the body mass in June. Body mass in June is the closest we can get to the body mass of a marmot that just emerged from hibernation. That happens in May, early May. So it's like a month after. The environmental driver that I'm measuring there is the spring temperature. And the spring temperature that basically influence or determines the food available during the spring when they come from hibernation. So we think that there could be two things going on there. One, they can get fodder during that time when there is warmer temperatures because there is more resources available. But also, once there is some resource available, marmots are changing their foraging behavior. So they start eating more so they can gain more weight before actually getting in the reproductive phase. So, so one, it's like behavioral / physiological, and the other one is about food, like actual resource availability, as you said. That's one part. And the other part was about the population dynamics, but I think I forgot already.
It's okay, you already answered my question. Thank you.
Oh, okay.
Then we have a question by Carolina, please.
Hi, my name is Carolina. I'm a PhD student from the University of Portugal. And I have a bit of a curiosity about the last experiment that you taught us about the zebra finches. And you put together groups of males and females that didn't know each other. But were there brothers and sisters among those groups? Or they were completely unrelated?
Okay, pedigree question. They totally unrelated. No, they come from a captive group. And even though in this captive population, the parentage among all individuals in the big group, it's very, how to say this? They take good care of not inbreeding animals too much, so they maintain the diversity and the a good amount of outbreed, like they prevent inbreeding basically in the population. So I cannot say, well, they were totally unrelated to each other, right? They do share some amount of genetic material, but they are basically differentiated enough to not be kin among them. If that's sort of the question. They are not sisters and brothers. Yeah.
Yeah, that was my question, because I was wondering, I don't know if that happens or not, but if they could recognize each other even though they didn't grow up together.
Great question. I think from my understanding, because I didn't really work that long enough, but from my understanding is that at some point they do not recognize roots. So individuals from the same roots, they won't recognize each other. But also in the wild, once they reach this dispersal time, they will all leave the colony. So it's less likely that they will encounter like close related individuals where they migrate. Yeah. So
Thank you. Great talk, by the way.
Thank you.
Then we have another question by Eduardo.
Thank you. Adriana, you open up with a couple of photographs that really reminded me of my trip to the savannas of Venezuela and very much to our field site in Argentina. I mean, the resemblance between those first few photographs and some of the habitats we see where we work are remarkable. But more importantly, I don't know if everyone in the audience could relate to those wonderful animals that you showed us first, the capybaras, which were supposed to be my dissertation research system. Yes. So I went to look for capybaras in Argentina, and then I had kids, and I ended up studying monkeys. But do you, I mean, have you, I know that you've done some work with the capybaras, and I just wanted, since I wanted to know, are there any studies that have studied them consistently? I'm very curious about that, and always hoping that I'll get started with some research on capybaras, maybe in my next life.
Yeah, well, we should join forces in that case in my next life.
No, you can do it in your, you still can do it.
I mean, in my next life, in like,
you can do it. We have them all over the place in Formosa, very accessible. I mean, I have had my kids sitting 50 centimeters from them, very habituated. Wow. But is anybody in Colombia studying their behavioral ecology?
No, in Colombia, no. I could. Oh well, there is a recent study about giantism in capybaras. So they study their genes basically to look for traces of why, or explaining why giantism, and relate that to humans. That was the most recent study from Colombia. And then I think I have to say that it's me, but that was some years ago already. And there is no like long-term study or a focal population that we follow or anything like that. Venezuela has done a better job on that. So I'm not sure if, I forget his name, Herrera. He's been studying capybaras for a very long time. And what I'm not sure if he studies the same group, like the same population, or if he changes according to the viability or something. So that's not quite clear from the papers, but that's the only one.
I thought of asking this question, which may be seen as unrelated to the specifics of your talk, because we have, I mean, a theme through the 5 years have been that many of us believe that it's necessary that we continue to extend the number of systems, and we continue. And I've always been puzzled why nobody has taken on capybaras. I mean, they don't move fast. They're big. You can find them large groups. They're plenty numbers. So the only challenge used to be water, which you clearly showed. So why do you think? I mean, what would hold you from getting into developing the capybara as a mammal system?
So in Colombia, what sort of prevents me to work with them is the accessibility to the places where you actually can study them safely, I would say. So we don't have like a biological station where, like Rumble, say, where we can put devices on them, or like have a research facility or something. And also to get to the place where I work with them, I will have to either take a plane to one of the cities in the eastern savannah. So I will have to fly to Villavicencio or Jopal. And then from there, I would have to take a bus to a little town. And then from there, I will have to hire a car that will take me in a 3 to 5 hour drive, depending to where I was going, to the middle of the savannas. And then I'll be there, no cell phone signal, energy or whatsoever. So I think that
logistics
are a key point there. So that's mostly what prevents us from doing long-term studies on them, I would say. But it's a really easy system to work with. And once you, I mean, if you know the people and also you learn how to trap them. I'm impressed in Venezuela that they just come into they just line up for the study. In Colombia, it's not that easy, and we tried, and they are very stressy, so they die if we try to manipulate them too much. I don't know if there is a physiological difference between Venezuela and Colombian capybaras, but yeah, our capybaras are not so into being manipulated or enclosed under any circumstance.
It's interesting you say that. There were some attempts, again, when I was studying my dissertation, a well-known Argentinian mammalogist, Jorge Kravets, who was one of my professors, he studied a captive colony. They were trying to breed capybaras in the delta of Buenos Aires. And I remember, now that you mentioned that, I remember him describing all the difficulties they had. And then there was somebody else, his name is escaping me, from Argentina, who did some population studies, hoping of doing some kind of sustainable harvesting, breeding. Interesting. Thank you. Maybe we should talk. We should talk about having a Colombia, Argentina initiative at capybaras.
That'll be great.
Thank you.
Okay, then we have another question by Gloriana.
Hi. So I was wondering also, Adriana, for the zebra finches, you explained that for the pair bonding study that it's still not published, right? So the way you measured relationships was based on proximity of the animals. Yeah. So how did you measure those relationships for the study where you gauged disturbance and the effect of disturbance on foraging efficiency? I didn't
No, that is good catch there. I didn't explain how I built the foraging association networks. So if you all recall the video, there was a piece of zucchini, and then individuals will come and forage on the zucchini piece. And I will call an association if individuals were around this zucchini piece. So basically we'll measure, again, this is doing scaling and standardization of measures and how pixels translate to actual distances and so on. But we will basically identify like the ring around the zucchini that corresponds to the rings made by the QR codes. So everything within that will be considered an association while foraging.
Okay.
Yeah.
Okay. Yeah. Thanks.
Okay.
So the next question is by Rebecca.
Hi, thank you so much, Adriana. Really nice talk. And I just have a question regarding the marmots and whether their social interactions and their social network structure actually changes as a function of them having given birth. So are their social interactions more diminished, or are they actually becoming more friendly with one another after they have babies? And do you know of any of that, or what would you expect?
Well, you know, that's a question that we've been trying to address, but well, not specifically that one, but it's a great question. But the problem with the marmots is that they are not very social, right? They don't interact that much. So the amount of data that we have to actually infer interactions in a shorter period of time is not enough. So that's why we have to clump everything into a single season. So we don't know that we don't have means to actually infer if they do change or not after the breeding season or the babies are born. And from my experience, I'm trying to think if they were more sociable. Maybe if Dan is around, he has more years of observations than I do, but I don't recall seeing a particular change between adults. Definitely, you can see some males become more, let's say, amicable with the babies. Males are not very amicable in general. They don't like too much, but yeah, we have some males, and this varies a lot within the males. And some males are really, really good fathers, and they just hang out with kids and with pups and let them play on top of them and so on. But even females, sometimes the pups will want to play and they will just like hit them and just like get away, go and play with the other pups. Don't bother me. Yeah. So yeah, sorry, I can't.
Yeah, that's okay. Thank you. How many pups in a litter usually? 1, 2, 3. I know nothing about marmots.
Yeah. On average, I think if I recall correctly, it's 4. They can go up to 8 in a single. Yep.
Okay. Yep. Great. Thank you so much. Nice.
Thank you.
Okay, it's not so always easy to keep an overview what's happening in the chat. So I just tell you, Adriana, there's many messages for what a great and wonderful talk it was, but that people had to leave. But I see that Leah and Lindelani are still here, so I guess there will be more questions coming. No pressure here, but it also gives me another opportunity to ask a question, even again with the risk that maybe you have answered or addressed it before, but I have to look at too many things at the same time. Probably you anyway cannot answer it. It's about this study, long-term data analysis by Arpad Oskul on the population dynamics in the marmots, where there's a sharp increase after the year 2000 or 2012, I don't remember. No, 2000 and then 2012, there's the sharp decline. And of course, this has been related to temperature, food availability, and duration of hibernation. Do you have any information from the Marmot project about another factor that might be important, that is predation, whether predation pressure might have changed also due to human impact over this long term? That could, because when I think about, of course, our small mammals are very much smaller than marmots, but I think that would be something that might have a severe influence, and it could change in the long term also.
Well, Carson, I have to say that I'm not sure. I mean, we would have the data to sort of answer that question for sure, because during observations, we have to record presence of predators around. And indeed, several of the questions that are happening or being answered in the Marmot project are related to predation. But I have to say that I have no idea if the predator has increased with time. I remember at some point we did have a fox that was getting super good at eating pups. And so basically that fox diminished the population in town. We call town where the actual station is, like all the cabins and so on. So marmots will basically build their hibernacula under the cabins, and then the fox will just sit there and wait for the marmots to come out, and then they will eat one pup at a time until they will eat everything. And there we had left like, I don't know, like 3 marmots after having like 1 marmot under every single cabin in that town. But yeah, I'm sorry, I have to say I'm not quite sure if that would be, there is a trend in predation risk. Yeah.
Okay, then thank you very much. I do not see any other questions in the chat here, which means I'm going to stop the live broadcasting on YouTube now. And of course, the people who are still around, you can still stick around here and can still ask questions. But this will then be the end of the live broadcast. And I would like once again to thank you very much for such a fascinating talk covering different taxa and present, current, past, present, and